Good morning, dear students. Today our lecture will be about the collagen biochemistry. The objective of this lecture is to list the main function of the skin and the component of the skin. In addition, we will state the biosynthesis of the collagen and its unique composition and the main function of this molecule. And also we will correlate clinically with a certain condition and disease that result from a defect in collagen biosynthesis. One of the dermatologists, giant dermatologist, was asked, "What is the true function of the skin?" His answer was, "What function the skin cannot do?" So the importance of the skin came from its being the largest body organ, as it's about 2 square meter in area, and also it is about 5 to 6 kg in mass, and because of its position as a barrier between the internal and external environment. The skin being in this position can provide the body with a barrier against the change in the temperature. Also, it is important to maintain the electrolyte balance, the fluid balance by preventing the excessive loss. It can provide a microbial barrier, a chemical barrier, and a mechanical barrier. More protection provided by the skin is by the UV radiation absorbing pigmentation system. In addition, it has an immune regulatory network which can sense tissue microenvironment for any foreign or abnormally expressed component. So this provides that the skin is very important in maintaining body homeostasis than previously was thought, and this is because of the skin multifunctionality. The human skin composed of 2 broad layers or tissue type, which is the outer layer, the epidermis, which is an external stratified epithelium, and the underlying is the dermis, which composed mainly of fibroblasts that can produce fibrous component and elastic component in addition to the ground substance. The dermis contains far fewer cells than the epidermis, but instead it composed of fibrous and amorphous extracellular matrix. The main cell type in the dermis is the fibroblast. The extracellular matrix composed of a protein, a glycoprotein, composed also of glycosaminoglycan and proteoglycan that we will study in the next slides. And the main protein and the most important is the collagen. Collagen is synthesized by the fibroblast, and it can give firmness and rigidity to the tissue. In addition, it contains another protein which is elastin. This protein can give the elasticity. So the extracellular matrix mainly composed of protein, glycoprotein, glycosaminoglycan, proteoglycan, and the main 2 protein is collagen. This is the major protein, and in addition we have the elastin. In addition to the fibroblast, we have many other type of cell. For example, the macrophage, the mast cell that can secrete histamine. The extracellular matrix was previously and wrongly thought as an inert ground substance only, but recent research have show that it has a multifunctionality. So the extracellular matrix, because of its vast majority of components that include the protein as collagen and elastin, the protoglycan, glycoprotein, and hyaluronic acid and other material, it gives a lot of function. For example, it's very important to regulate the cell migration, proliferation, the survival of the cell. All is controlled through signal from the extracellular matrix. Also, because of this different material that is found as a part of the extracellular matrix, it has a filtration barrier function. And also it allows the exchange of certain material and water and ions between the two different layers. And also because it contains the collagen, which is responsible for the firmness and rigidity, so it is important for the stabilization of the tissue structure. We say that the main cell type is the fibroblast. It can repair and degrade different components. So in case of any trauma to the skin, whether mechanical or by UV or by physical, it can repair. It have extracellular matrix have a repair function, and also it has immune process through a hosting of different cell in addition to the fibroblast. Also the mast cell that contain the histamine and other cell and other signaling molecule. For example, the interleukin and cytokines also is found in the extracellular matrix. So it is very important in the immune process and as a player in inflammation. The most important protein in the extracellular matrix is the collagen, and it forms about 90% of the dermal protein, and also as a weight it forms about 75% of the skin weight. And the collagen word come from the Greek word colla, which means a glue producing. So it acts as a glue that connect different parts. Different types of collagen are found. About 25 collagens exist, and half of these are found in the skin. It can function in a diverse range of situations. Collagen can provide tissue integrity, so it's important when we have a damage to the skin. It's important for a tissue repair. Also, it's important for adhesion between the different molecule, and also important for migration of the cell. The main function is to provide the structural support because it's rigid and have a resistant ability when it is stretched. كولاجين طبعا مو فقط موجود بالسكن، وإنما موجود أيضا in the ligament, in the tendon and in the bone. About 28 various type of collagen are found, and بالسكن predominantly we have collagen type 1 اللي يكون تقريبا 90% of the collagen in the skin, and we have a collagen type 3 اللي also about 8 to 11%, and 5 which found in a percentage of 2 to 4%. And this can change when we have the proportion of these different type can change according to the situation. For example, in certain repair condition, one type predominate over the other type. Because of the dominant role of collagen or position in the skin, so it is not unexpected that it have a diverse range of function. So as we say, it is mainly for support function because of its rigidity and firmness, strength. So all this give a supportive role to all the structure found nearby. Also, it can give attachment site for the cell. And this attachment site can help in the differentiation of the cell, the proliferation of the cell, and the migration of the cell. And in addition to the supportive role and the cell attachment site, the collagen can interact with the cell either by receptor or by protein or by proteoglycan. So it can react with the cell either by receptor اللي هي integrin receptor or by adhesive protein اللي هي laminin and fibronectin or by the proteoglycan. Since a collagen is a protein, so it is composed of amino acid, and it have a special arrangement. It is arranged in a triple helix. So triple helix means that each molecule composed of 3 chains that are twisted around each other. The most common amino acid and the smallest of all of the amino acids that participate in the structure of collagen is the glycine, and because of it is small, it can provide tight configuration to the collagen molecule and allow it to handle the stress. Collagen is mainly synthesized by the fibroblasts, which are specialized in this function, and the synthesis of the collagen, a part of the synthesis, some steps occur inside the cell, and the remaining steps occur in the extracellular space or tissue. So part is intracellular and part of the synthesis is extracellular. We have about 25 genes that code for the synthesis, and the amino acid that is found in the collagen have a specific composition. We have 5 of all of the amino acids are dominant, which are the glycine, proline, hydroxyproline, lysine, and hydroxylysine are dominant. These 5 amino acids dominate the structure of the collagen. In addition to the special triple helix arrangement of the collagen, we have also a special arrangement of the amino acid in the collagen structure. فإنه it happen in repetitive tripeptide sequence. Every 3 amino acid together, we call it tripeptide sequence, repetitive tripeptide sequence, every 3 amino acid. The main composition is glycine XY. The first amino acid is glycine, then the X could be anyone, but any amino acid, but it is mainly either proline, mainly it's proline, and Y could be either hydroxyproline or hydroxylysine. إذن بالإضافة إلى كونه هو عند triple helix structure composed of 3 chains wound together, each chain also have a specific amino acid sequence composed of tripeptide 3 amino acid اللي إحنا نسميها glycine X1 XY. الجلايسين معروف هو amino acid، هو قلنا the smallest one، والX it could be anyone but mainly هو proline، والY عادة يكون hydroxy proline or hydroxylysine. فهذا specific structure for the collagen. And الcollagen fibril composed of 3 polypeptide units، وأيضا هو إنه occur in 3 polypeptide unit, and we call it tropocollagen. Between the polypeptide strand of the triple helix, we say that the collagen are arranged in a triple helix, and between these polypeptide strain, we have a bridge are created, and these bridge help to link these 3 peptide together. So we have a bridge between them, and this bridge is created mainly by the lysine, so that it can give more strength to the collagen. And the cross-linking of tropocollagen will give the strength and the toughness to the collagen. This reaction that involved the cross-linking between and formation of bridge between the polypeptide strand of the collagen is by the enzyme lysyl oxidase. So this is an important enzyme, lysyl oxidase. It catalyzes the formation of a cross bridge between the polypeptide strand and the triple helix of the collagen, and this cross link is very important to give the strength to collagen. And this process continues throughout the life, and the cross link usually accumulate in the in the skin, and it is responsible for the loss of elasticity that we see sometimes in aging. The biosynthesis of collagen usually occur in the skin, and also collagen is found in the bone and in the cartilage. So in the skin it is synthesized by the fibroblast, in the bone collagen is synthesized by the osteoblast, and in the cartilage the collagen is synthesized by a chondroblast. The first step is the synthesis of the pre-pro-collagen polypeptide, which is the precursor for collagen. So the precursor molecule is called a pre-pro-collagen. And we have a certain sequence of amino acids that are called signal peptide, and this precursor molecule that is called pre-pro-collagen will allow the entrance of the molecule into the endoplasmic reticulum. Then the pre-pro-collagen will be cleaved to form the pro-collagen. تحول من pre-pro-collagen إلى pro-collagen. Then we have the post-translation modification that involve certain amino acid in this molecule. We have two processes. We have the hydroxylation and glycosylation of two amino acid mainly, which is the proline and lysine. In the pro-collagen molecule, this is a post-translation modification. Then it was first synthesized as a pre-pro-collagen, then pro-collagen, then there will be hydroxylation and glycosylation of the proline and lysine amino acid in this molecule, and then a bond is formed between three pro-collagen chains, and after forming of the bond between them, they will be twisted around each other to form the triple helix molecule. This is all these steps are formed inside the cell. We have the pre-pro-collagen, the pro-collagen, the hydroxylation and glycosylation, formation of the bond between the three chain, and twisting around each other to form the triple helix molecule. This step أسميها the registration. All these steps were done in the inside the cell. Then it will be secreted to the extracellular matrix, the formed molecule, which is the procollagen, and then there will be more modification in the extracellular space. There will be two enzymes, the aminoproteinase and the carboxyproteinase enzyme. They will remove the extra amino acid, the terminal amino acid, and then the pro-collagen, it will mature and form the collagen. Then outside the cell, we have the step of removing the extra terminal amino acid by the procollagen aminoproteinase and carboxyproteinase, and formation of the mature collagen molecule. Then the collagen it will form fibrils اللي هي smaller than the fibers, and then it will form fibers, and these fibers by covalent cross link it will be stabilized. This is a diagram showing the steps of the collagen synthesis. First of all, we have the activated fibroblast. There will be the gene. A gene will form coding for alpha chain. Then after transcription and translation, there will be formation of a tri molecule, and then formation of the precursor molecule, which is the pro-collagen, and then after that, by removing of the extra amino acid at the terminal end, the formation of disease are by the carboxypeptidase and aminopeptidase, the formation of the collagen, and then the collagen will form fibril, and the fibril by cross-link will form the fiber of collagen. Many enzymes are important in the collagen biosynthesis, and these enzymes require cofactors. These cofactors include one of the major vitamin is vitamin C. In addition, also it requires iron and copper and other cofactor. Vitamin C play a major role as a supportive function and a cofactor for the enzyme lysyl hydroxylase and pyrrolyl hydroxylase, which are very important in the collagen biosynthesis. Patients who have vitamin C deficiency have a lot of presentation, as weakness, poor wound healing, may present with anemia, gum disease, and general fatigue. But one of the manifestations that is found in the skin is perifollicular hemorrhage, in which we have a bleeding and the follicle of the skin are found with perifollicular, which is small hemorrhage around the follicle, and the follicle will be plugged with the keratin. So vitamin C is very essential for a normal collagen bio. The degradation of the collagen or the breakdown of collagen is part of the remodeling of the tissue. Usually there are many pathways and steps that guide the degradation, but mainly either it's occur intracellular by the phagocytosis, or outside the cell extracellular by the enzyme matrix metalloproteinase. As we said, this breakdown of collagen is part of remodeling of the tissue, but sometimes we have an extra factor that increase the degradation of collagen. For example, the exposure to sunlight excessively, the UV light, and smoking, bad habits, all these will increase the breakdown of collagen. The sign of increase in the breakdown, abnormal increase in the collagen, is loss of the elasticity, appearance of a wrinkle earlier, and the saggy skin. The abnormality that are associated with the collagen, we have either a decrease in the production of collagen, or we have increase in the production of collagen. In case we have decreased collagen production, we will search for the cause of this decrease. Either we have acquired condition or genetic condition. In a genetic condition, the most popular and well-known syndrome that is associated with the decrease in the collagen synthesis is the Ehlers-Danlos syndrome, and another one is the osteogenesis imperfecta. The acquired disturbance in collagen occur because of deficiency in certain substance that is required for a normal synthesis of a collagen. For example, vitamin C, that is a cofactor for the enzyme that involved in the biosynthesis of collagen. So when we have a vitamin C deficiency, we will have decrease in the production of collagen and the associated abnormality with it. Copper deficiency also is associated with disturbed collagen synthesis. The effect of this deficiency could be either it affects the biosynthesis, or the arrangement, the proper arrangement in a triple helix and the twisting around each other. Sometimes the post-translation modification, which involve the removal of the amino acid at the terminal of the molecule, may be affected. Any process that involved the normal production could be affected by decrease either the acquired or the genetic defect in the production of the collagen. The increase in the production of collagen usually is seen in case of fibrosis. The manifestation is in a fibrosis, as we know that the collagen is not only found in the skin, sometimes maybe also found in other tissue. So we have abnormal increase in the collagen may lead to fibrosis, for example hepatic fibrosis, which is the cirrhosis, and other tissue also may be affected abnormal and etc. Formation of fibrosis during skin healing after a wound also will lead to abnormal appearance of the skin. Ehlers-Danlos syndrome is an example of an inherited defect in collagen synthesis. The cause is genetic, so there is defect or mutation in the collagen gene. There are 25 genes in collagen biosynthesis. In this syndrome, we have the defect is either in coli I1A1 or 1A2 or 3A1 or col5A1 or col5A2. And this results in when we have defect in or mutation in this gene, and according to the mutated gene, we may have defect in the lysyl hydroxylase, maybe in the procollagen and proteinase, causing them to form an irregular collagen fibril. They became abnormal and thin and irregular. So the end result is formation of collagen fibril that are irregular, thin, abnormal and weak. These different mutations may affect either the structure of the collagen or the amount produced of the collagen, or sometimes the processing, for example the post-translation modification of a collagen, or sometimes even there is defect in the protein that act with the collagen. The interaction between the collagen and other protein may be affected by this syndrome and the mutation that occur in it. The end result of this mutation and defect in the collagen biosynthesis is the patient may be presented with weakness in the skin, in the bone, in the blood vessel, in the organ, because a collagen is formed or is part of all of these structure. And the main characteristic is skin hyperextensibility. We can pull the skin to an extent more than the normal, and a joint dislocation because of the collagen. Normal function is to provide the rigidity and strength, the support to the joint. So if we have weak collagen, the support will be absent and the joint will be easily dislocated. The tissue generally will be fragile because the collagen gives a support to the tissue. In addition, there will be poor wound healing because collagen is a very important part of the extracellular matrix, and it is very important in tissue remodeling, important for the tissue migration, proliferation and differentiation that occur. And one of the and since the collagen is part of the part of the dermis, and it is one of the and the dermis is part of the skin. One of the main function of the skin is to provide the repair function. This picture shows the presentation of the Ehlers-Danlos syndrome, EHS syndrome. We can see that this picture shows the skin hyperextensibility of the skin, in which we can pull the skin to an extent more than the normal. And this diagram or the figure will illustrate what is the defect in this syndrome. The patient will become prone to injury. They have GIT disorder, fatigue, chronic pain because of weakness of the ligament that support the joint, and soft skin also be affected. There will be many bruises, and because of defect in the healing. Hypermobile joint, because we say that the collagen is part of the ligament that support and the tendon that support the joint. So weakness in the collagen need to weak in the support of the joint. They became very hypermobile because there is no stiffness and that was provided by the collagen. And there will be early onset osteoarthritis. Osteoarthritis هو فان. Why? Because also there will be weakness in the supporting tissue and defect also in the cartilage, in which collagen is part of it. This is example of the hypermobility of the joint. Usually the normal person cannot move the joint to such an extent, while because in this syndrome, because of the weakness of the collagen that's part of the supporting tissue, this patient can perform such an action with his joint. And thank you.